I'm going to open up something to share with you, remind you where we started for tissue, nervous system tissue back in lab. So the slide that you saw, I used to find, say, the nucleolus, which I can point to here at the end of the needle. But inside this slide, what I want you to be reminded about is the idea that you're going to have these excitable cells and be surrounded by non-excitable cells, and be called glial cells here. And that's pretty much the only thing we tell you about the nervous system when we introduce the concept of excitable tissues, right? So remember, we have epithelia, we have connective tissue. They are non-excitable, even if they do have resting membrane potentials. Muscle cells, cardiac, smooth, and skeletal, which you looked at last night, as well as neurons, are highly excitable cells. And most of the work of chapter 12 is figuring out, in this case, specific types of flattened multi-synapsing neurons regulate a collection, interpretation, and then subsequent communication of information electrically, such that ultimately the chemical signals, the acetylcholine, you guys have been thinking about it, the neuromuscular junction, that is a chemical signal that's initiated, its release is initiated by a change in electrical activity. So that's pretty straightforward in terms of, by straightforward, of course, I mean, as simplistically as possible, thinking about the idea that we have, one, a nervous system, and then two, what I'm hoping people are remembering is I've tried to use the lower motor neuron as the tool to remember how it is that information can activate skeletal muscle tissue. And one of the things I wanted people to do last night is wrestle with this idea that if we assemble, say, the vertebra of your spine so that we could actually run the, I'm going to take a look at a specific portion here, right? So if here's the body, here are the transverse processes, transverse foramen that are going to allow for the movement of the actual nerves. That when you read the book, you're going to realize a nerve is not a neuron. A nerve is the sum of the axons of neurons that could be communicating information in either a afferent, meaning toward the central nervous system manner, or efferent, away from the central nervous system manner. So Mother Nature needs to create a system where information can be collected from the outside of the body and then sent to the inside of the body. All right. And here's a really good way to think about this. If these are the thoracic ribs, say the rib would be here, cartilage, there's the sternum, here is the central portion of the body of a vertebra, and here's that transverse process again. You can see these spaces that are opening up in between, okay? And this intervertebral foramen is the one we're actually looking at, right? So the intervertebral foramen allows for nerves to pass in and out, okay? So that's what I thought it was. Hold on a second. Can't actually see one on this, sorry about that. So by using, what I'm thinking are the important pieces of the lab we've covered so far, the labs that we've entertained in terms of, if we're thinking about how Mother Nature is going to use, say, the nervous system to collect information. And all I keep saying is it is going to collect and then sort of interpret and then respond to it. And this is an important idea, right? Because let's just jump right to it. Okay. And what I'm hoping you're seeing here is that you can see we're entering, this is the lab book, right? And what I'm doing is first I'm going to introduce the parts of a neuron, which if you've started, you can see the telodendria are now the distal end of a neuron. And this is a specific type of neuron. It's a multipolar neuron. There are going to be three major types, okay, that you're going to be aware of. Let me start adding my face back into this, right? Okay, so now that we are in this, here's the multipolar neuron. The teledendria are these things we're going to make synapses with, and that's a word I've used a few times now, right? The synapse that's formed at a lower muscular junction is the end of one of the axons of a neuron. And in the case of lab, that was that lower motor neuron. How cells collect information are these structures that are called dendrites, okay? And dendrites look like a root system, but how they function is they're decorated with different receptors to detect specific types of signals. So unique neurons pick up unique signals that will then be converted into electrical information. And that electrical information will then be converted into chemical information down here at the teledendria. Okay, so how we use that, okay? We move now into, say, the soma or the cell body. This is going to be covered with a neurolemma, right? And this is the axolemma, would be the membrane surrounding the axon. And these structures decorating the axon in blue, these are going to be called myelin sheaths, and they are part of that glial non-excitatory system that's used to support the actual nervous system itself. Okay, so this is those myelin sheaths I was telling you about just a moment ago. And over here, a very important piece inside of chapter 12, at least in terms of functionality, right? It's going to be, what is an axon hillock? Why is it important? And really what it's doing at the end of the day, okay? And this is just me saying to you, hey, this is going to be someplace where we are going to sum the activities of all of the actions that have been collected from the dendrites in the cell body. And you're asking yourself, well, what's going to be summed? Well, what's going to be summed are going to be the excitatory events, we're going to call positive, inhibitory events, which we'll call negative, and these will be depolarizations, DEP, and these will be hyperpolarizations, HYPER, right? And you'll notice the key word here is polarization, and that means that you understand that the charge at the membrane is now set up specifically for us to either depolarize it or hyperpolarize it. And that's the work of chapter 12. It really is. That is the hard stuff, because unless you understand what a resting membrane potential is, then you don't understand that by the information that's collected here at the dendrites, that's going to now change the polarization. All of that has to be summed here at that axon hillock. And that's the basic mechanism of chapter 12, because if becomes a series of logic statements. If go, then nothing stops it. Everything will move electrically along the membrane while it's still undergoing its normal physiology, transporting what it needs to transport. When you get into like what is axonal transport, you cannot confuse that with what's being signaled at the membrane. And then the release of a neurotransmitter will happen. Just put NT here, because inside chapter 12, we introduce the many types of neurotransmitters, but is by no means an exhaustive list. Okay.
I have a question.
Fire away, Jen.
The teledendra, is that the part that we saw on the muscle cell?
No, that's actually, those are so they're called teledendria here. Okay, and I think the piece that I would say is that the lowest piece down here is what you were looking at, right? Those would be the synaptic M bulbs.
Yeah, that.
They put the whole teledendria together, saying that, you know, this is going to be the branching part at the end of the axon. How does that sound?
Understood.
Okay, cool. So yeah, so if I was pointing at that synaptic M bulb, you know, I'd expect synaptic M bulb. You could give me teledendria. I wouldn't probably mark it wrong, but the teledendria is the sum of all of those things, right?
Okay.
Okay, so let's clear that. Clear. That's cool, fantastic. So, and this gets into right here, if you think about, let's get rid of this first. In this figure I'm about to show you with that synapse, this fits in nicely with how it is we are signaling, say, a neuromuscular junction, except we're not at a neuromuscular junction. Notice the part here is the dendrite, so it's another neuron. But here's the end of one, so this is a synaptic M bulb. This is the axon terminal. which is called the synaptic end bulb. And the space between them is the synaptic cleft. Cleft just means space. And what's going to happen is the charge is traveling from the resting membrane potential. Notice they're trying to demonstrate these are different channels. Notice they have little sort of red balls here for sodium voltage gated channels, and they have the green ones here for calcium voltage gated channels. Well, what happens is the charge opens voltage gated calcium channels, just like we saw in the lower motor neuron. That calcium being brought in allows for neurotransmitter to be released. And in chapter 12, they tend to talk about axonal, in this case, axon terminal to dendrite, dendritic signaling. So the neurotransmitter binds the receptor here, and now you're going to cause some form of potential change. Notice here, we're calling it a graded potential. Okay, your textbook is going to start calling them different names, but for this evening, at the dendrite, at the cell body, we are interested in the actual graded potentials. So now you were asking about the neuromuscular junction. So here's the telodendria. Those are the processes, but the actual terminal piece where you're actually synapsing. Notice where I said you have one per muscle fiber. You could actually count that and verify that I didn't lie to you, but there's one nerve that's traveling to that area. The telodendria are now separating so that they will recruit individual muscle fibers as needed. Okay, so that's a neuromuscular junction, and this is inside the nervous system here, where all of the same responsibilities have to be executed. Earlier, this is the same figure I was showing you earlier here, where we were looking at, this is the nucleolus here, this would be the nucleus, and these would actually be the dendrites that are moving away from this one cell body or soma, right? We don't really care necessarily inside, for us, what the individual glia and neurons look like inside of the different parts of the body, but for the purposes of class, there's going to be 5 different types, well, 4 major types, right, of glial cells. And the significance of that is going to be that if you have an ependymal cell, if you have an astrocyte, if you have a microglia, you know that those are not excitable cells inside of the nervous system. In fact, they tend to have the responsibility of keeping others out of the central nervous system, and why that's so important. Okay, and this is where we're headed with this. Okay, so inside the central nervous system, we'll have ependymal cells, astrocytes, oligodendrocytes, and microglia. And the significance here is from E, A, O, and M, you know you're inside the central nervous system. You're in the brain and the spinal cord. Peripheral nervous system, when we use the word Schwann cell, it's going to make myelin, just like we see these oligodendrocytes, but Schwann cell means you're outside of the central nervous system. Okay, so that would be those neurons that are traveling to the skeletal muscle tissue, or those neurons that are traveling to the organs of your body, right? Now, each of these are developed in my lecture. What does an ependymal cell do? It filters and then circulates cerebrospinal fluid. It's responsible for actually making astrocytes. These make up that blood-brain barrier such that the ependymal cells now have materials to transport. Oligodendrocytes are going to make myelin, and that myelin is going to be responsible for the differential electrical conductivity at the axons of neurons. Okay. And microglia, these are white blood cells that function inside the central nervous system. We didn't think this happened up until about 5 years ago, 6 years ago now, right? Where they actually go through your entire central nervous system, but they know exactly in a central nervous system specific way how this would actually function, right? Whereas inside the peripheral nervous system, we'll talk primarily about Schwann cells and satellite cells, which support neurons inside the ganglia, which are going to be groups of neurons that are outside of the central nervous system. Now, I don't want to go through every single piece of this. In fact, all I really want to do is I want to get first to our models we're going to look at this coming lab on Thursday, right? So let me see if I can just skip down all the way through this. Wham, wham. We had that earlier. Nope, Let me move back to the skeletal system. That's what I want to do. Sorry, I'm just going to blast through this quickly and pull up the images I want to look at. Muscle, right? There we are. Cool, fantastic. So this is a model that I've been using since the very first day of class, right? And what you have here is last night you saw this is your frontal cortex. This is why you need sleep. But when we look at, say, the central nervous system, that is the brain and the spinal cord, right, which goes through these vertebral structures, but those structures that are coming off that are going to be the nerves, those nerves are part of the peripheral nervous system. So the cranial nerves and the peripheral pairs of nerves, these are not part of our central nervous system. It means they're going to form a synapse someplace, and we'll figure that out later. But what I want you to see here is this is a reflected surface. You pulled the patient's bones out of the way. You pulled their organs and organ systems out of the way. And what you've done is you've removed the meninges, which is the dense, irregular connective tissues that are surrounding the brain and the spinal cord, so you can see that this structure actually comes to an end. This thing is called the conus medullaris. We then put this thing that's called the horse's tail or cauda equina, and these are the axons of neurons, which become nerves that travel to the distant locations inside of your body. Remember, your classmates were asking about the sacrum and how it is you can actually have herniated discs and why it is your legs go numb. Inside of this, we have to anchor it, and we use the denticulate ligaments as well as the filum terminale, and the fact that your spinal cord attaches to your brain to keep it all in place. Okay, so how does Mother Nature do this at the individual vertebra? This is what you'll see tomorrow on Thursday inside lab, and this is color coded for you, but this is the bone. So this is the bifid. So we know we're in the thoracic portion of the spinal column. And these will be the facets that are allowing for articulation between the actual transverse processes. And then here's the body of a vertebra here. Inside this space, let me see if I can make this bigger for us. I can. No, but I can't. Let me do that. Sorry about that. Okay. You can see that we are going to have this connective tissue layer, which are going to be called the meninges, and this is going to be the dura mater. Okay, and then you'll have the pia mater, and you'll have an arachnoid matter. And Mother Nature assembles them in a specific order. The easiest way to remember it is DAP, dura, arachnoid. Then you'll have a subarachnoid space where we'll have cerebral spinal fluid, and then the pia mater. The pia mater always sits on top of the nervous system tissue, both outside and inside. This thing is called the central canal. So when you're looking for that in lab tomorrow night, that's the sort of thing that's really important. How do we separate the central nervous system from the rest of your body? And it's going to be the meninges that are responsible for that. So where do I want to go with this? Well, if Mother Nature has that in place, notice that she has some additional pieces of information on the outer portion. Like if we're taking a look specifically now at what are the structures within the spinal cord. I'm sorry, though, it's getting dark outside. You can see that we have specific names that are for the horns, and you'll see dorsal, okay, as well as ventral horn. So ventral will be the anterior surface, dorsal will be the posterior. You'll see the columns are areas that are whitish in nature, okay, or in this case, gray. And the significance here is this, is that they either have a large amount of myelination or they have a very small amount of myelination. All of these columns, large amounts of myelination. All of the horns, very little myelination. And to travel between the right and the left sides of these, we would use this thing that's called a commissure. Okay. Outside here, outside the bone, where we've traveled out, we're now in the peripheral nervous system. Our Remy communicants and our sympathetic chain, these are part of our autonomic nervous system. We don't think about those for a little while, but for us to get signal into and out of the central nervous system on both the right and the left sides, we have these ramus, and these ramus allow for things to either go to the front of the body or to the back of the body, right? If you watched my earlier preparations for this inside of the test 3 folder, right? You'll see that I put together a series of asynchronous talks, and I talk about what it means to create this thing that's going to be called a dermatome, right? And that dermatome becomes a very important idea when we think about how sensation occurs. So this would be in red now, just to sort of reinforce what I just showed you. These would be unmyelinated structures, and this sort of like teal blue color is the myelinated structures. Now, for right now, if you can understand that, that's going to be very important, because what Mother Nature is going to do is she's going to say, well, why would I do this? Why is this so important for you to understand? What I'm going to do is I'm going to show you how she utilizes some of this, right? So this looks very familiar, right? You can see the yellow lines. You can see where the axillary versus the radial versus, say, the medial versus the ulnar nerves, okay, are located, and why it's so important to make sure you see the difference in them, right? So here's the brachial plexus and the brachial nerve that is traveling out here. And notice it's underneath the armpit. So that's why when somebody wrote on their practical, the axillary nerve, notice it is superficial. It wouldn't be underneath here. Okay. Now, the significance, of course, is these are just branches of names. And what I wanted you guys to do is recognize that inside your textbook, sometimes they just told you which don't travel where you think you're going to travel to, okay? And that's fairly important when you're trying to map what all of this stuff is doing inside the body. But for right now, let's think about just say the lower portions. If we take a sciatic femoral, these are large structures that are traveling. Notice this is right alongside the bone and traveling into the bone itself. In some cases, pudendal traveling on the inside face of the thigh, okay, and the sciatic traveling all the way down the length of the leg itself. And this is just descriptive. I mean, it really is. It is, hey, this is the name of the nerve. This is where it's traveling to. But last night, I wanted you to go from, look, here's the muscle. Here's where we're going to find it. Here's the origin, the insertion. Here's the joint that it crosses over. And now the nerve that's going to activate it. So the femoral nerve, right, would be responsible for activating which muscles, right? If you put your table together, maybe it covered it and maybe it didn't. So let's think about how we build nerves, because this is at least going to be familiar, right? And that's familiar is not a bad thing sometimes, right? So if a nerve is those yellow things that are traveling all throughout the body, notice it has the epineurium surrounding it. You have a perineurium surrounding fascicles, and the fascicles are holding the individual myelinated neurons. And in some cases, only holding unmyelinated neurons, and they travel together, right? The whole point is that sometimes you'll have an 18-wheeler traveling down the highway next to a Pinto, which is traveling next to, I don't know, throw in your favorite car type, okay? But the point is that what Mother Nature does is she makes these conduits that are surrounded by connective tissue, and that dense, irregular connective tissue, at least down to the endoneurium itself, because the endoneurium is most like areolar connective tissue. The perineurium and the endoneurium, these are dense, irregular connective tissues that are used to support different forces, to withstand the different forces surrounding them. Okay. So even though I said, right now we don't need to think about what are the 12 cranial nerves, because this is for chapter 14, but for lab tomorrow, what I want to do is I want to introduce this idea that we have a series of nerves that are going to work with the peripheral nervous system, right? And we use them primarily, believe it or not, we use them as neuronal checks. Something goes wrong, you've injured your head. The idea, of course, is that by using a series of quick cues, you can figure out which damage has occurred. This lab book makes a big deal out of the landmarks for figuring out where you are, but really the simplest thing to do is the lowest number are towards the anterior portion, the highest number are towards the spinal cord. And if you can remember that, you're going to be fine. Okay. Now, if we think about what they are, I'm just going to put this up here just for you guys to take a look at, right? And you're going to see that even in this figure, let's see if I can make that bigger for us. Fantastic, we can. Okay. Cranial nerve, we ask you guys to learn the name and number, right? So you can see that olfactory, optic, ocular, motor, trochlear, trigeminal was the big one we saw in lab last night. Inducens, facial. If we think about like the most, I would say the most important, or the way I tend to think about cranial nerves, if I cut them, what happens to the patient? Well, vagus and glossopharyngeal are the two most important cranial nerves, right? If I cut these, you die pretty quickly, okay? Maybe hypoglossal will be next, okay? But you'll see that, if I cut your olfactory cranial nerve, yeah, you won't smell, okay? I mean, literally, you won't be able to smell. But optic, yeah, you can't see, but you're not going to die. You may not die in a way that your normal physiology would stop. Okay? So to close this up and just maybe open it up for questions, the last thing that I want to cover inside lab tomorrow goes, so if those are the nerves, we need to think about what are the different parts of the brain and stuff like that. So let's get to that. Okay. Voila. Okay. So here's the actual cortex of your brain, right? And you're going to notice something, that they map the bones of the brain, right? So here's your occipital lobe, here's your temporal lobe, okay? Here's your frontal lobe, here's your parietal lobes, and then here's going to be your precentral gyrus and your postcentral gyrus. Remember, last night we had these color-coded differently. Somebody's blue and somebody's red. Why? Okay, between each of these things is going to be your gyrus. It's the central sulcus, is the dip between the gyruses. So the little depression is the sulcus, right? Okay, but our postcentral gyrus is where all that sensory input's coming from. Go ahead, find that one hair on your arm again. Notice it's going, you feel one sensation. And what's happening is it's going to one part of this entire cortex. And then for you to move that voluntary, remember your classmate who threw the pencil at me yesterday, that voluntary set of motions had to activate a series of steps, beginning here at this precentral gyrus. Okay. So now what happens, of course, is that if this is where things are happening, you're going to have a motor association area and a sensory association area. And that's more for your psychology class or a neurobiology class. But for right now, we don't tend to concern ourselves with it. Okay. Sweet. Now, what I want to do is I want to even make it more simplistic than this. Okay. Because when we take a look at, say, the diencephalon of the brain, which is going to be this portion here, this is going to be the thalamus, the hypothalamus, and the epithalamus. And that's very important because without any of this, you don't function as a living human being, right? So you'll notice here, they go for the lowest portion being the medulla, and you're going to work your way up from there. It's from the medulla to the midbrain, okay? Yay, pons, respiration, okay? So you can, you're going to find these in the models tomorrow night, right? This is not terrifically difficult. In fact, I would say it's not difficult at all. Okay. In fact, what I'm trying to do is find one summary, but you can't really see summary anywhere on these. Not true. Okay, cool. So we call these lateral structures, but because they're lateral structures, we can use them to orient ourselves. Because look, here's the lowest portion, which means that would be the fill in the blank. And here's our fill in the blank. Here's the posterior structure, okay, that's going to have its own referred to responsibilities for balance and movement. And each of these are what you're aiming for. And what I'd like you to do is just think to yourself the most prominent orientation point here is this thing that's called the corpus callosum. And the corpus callosum is now going to allow for communication between the hemispheres of the brain itself. Okay. Now, how do we actually make that cerebral spinal fluid? I go into this in lecture, but these structures are these things that are called the chloride plexuses, and I'm going to have you look for those not only in the models tomorrow night, but also in your dissection, right? And your dissection is important because you actually get your own little brain and you get to cut it up into a million little pieces, but not hopefully so many pieces that it's not useful to you. Okay, and let's now add back in how it is the actual meninges are going to function, because what's going to happen is blood travels to those chloride plexuses, you create cerebral spinal fluid, And then what's going to happen is you're going to drain waste into the dural venous sinuses, right? Which is going to collect all of the waste and then return it back to your circulatory system. Okay? So it's a very important idea that Mother Nature protects you in such a way that she'll allow you to now remove waste from the highly active metabolism of your neurons inside of your body. So the last two things that are in the lab tomorrow night are your eyes and your ears. And I'm just going to sort of introduce basic eye structure here for you. So here's your basic eye. You're going to have, say, a lens, this yellow-orange structure back here. That's actually your retina. And the sum of all the information that's collected travels out this optic nerve, which is cranial nerve number 2. Well, that's your retina. This is your vascular tunic. And in white outside here, this is your sclera, or your fibrous tunic. And what Mother Nature does is she puts liquid connective tissue inside here called hyaluronic acid. Remember in lab, I said if you push into somebody's eyes. And what's happening is you have a series of suspensory ligaments that are holding this lens in place, but they're changing the thickness. So depending upon what you're focusing on and where your light's coming in through the outer portion, right, and how much light we let in is the difference between how many neurons are stimulated through photoreception. How does that work? Well, it's above our pay grade this evening, but photoreception is going to come here, and we're going to activate those photoreceptors, and then we move into the actual ganglion cells. So you actually collect information the farthest position back in that retina, and then it's sent through a series of layers. And those ultimately, it's going to be collected into the axons of the cranial nerve number 2, right? So like I said, it's a little bit above our pay grade for right now, but how it works relative to, say, the ear, which is what we're going to go to now, is here's the external portion of your ear. You're listening to me. What's happening is sound waves are hitting this. They're being pushed into this external auditory meatus. They're going to hit that thing there. That's called the tympanic membrane. And what's going to happen is that information now travels through a series of bones, incus, malleus, and stapes. Okay, the correct order is malleus, incus, stapes, right? And we're going to move fluid inside of this structure over here that's called the cochlea. Simultaneously, though, because you're working constantly against gravity, you have this vestibular portion. So that's why when you look at the cranial nerves coming out of the ear, you're going to have the vestibular cochlear portions of those, right? So even though I'm not going to discuss it, all that much longer here, what I want you to see is that how Mother Nature makes this work is, here's this picture right here. Here's the cranial nerve. What's going to happen is if this down here, that's actually your tympanic membrane. These bones move, you move the fluid inside this space, and inside the actual cochlea here, we move a fluid, and that fluid will now allow us to actually hear. And how it works is you actually, so this is inside that cochlea, right? You're going to have these different compartments, scala vestibule, scala tempia, okay? And you have this vestibular membrane. And what's happening is the fluid's moving, and you see this thing is called the tectorial membrane. Well, what it's doing is it's pushing on those hairs. And this is a mechanical receptor. So no different than the hair on your back of your hand, you hit that, it now is going to travel through the specific cochlear portion, the vestibular cochlear neuron, right? Traveling, axons traveling now through the cranial nerve to actually get to the brain for you to interpret that. So in many ways, it's not any different than the hair you have on the back of your arm. You're detecting it. But the interesting thing is this. The frequencies that you hear are determined by the tectorial membrane pushing on those hairs such that you're activating this thing that's called the organ of Corti in a unique way for the range of sounds that you could hear. Right? And that's pretty, yeah, go ahead.
Is that why children can hear a different range than adults?
Yeah, especially if like we went to like heavy metal concerts when we were kids. or we drive in traffic on 95 all the time, yeah. So, the hair grows back more quickly and all that sort of stuff, right? Meaning inside the ears, right? So, I'm finishing up here, but the idea, of course, is that this is in many ways no different than how we introduce sensation. And in many ways, it's no different than how I want you to think about that. If you have these peripheral receptors, all the information has to travel in. But in this class, we don't really teach you how Mother Nature interprets all of it. We just say it's going to go to a specific location. So in the case of, let's say, chapter 17, which I'm going to cover in the lab tomorrow, right? I'm going to say, well, look where it's traveling to. I'll open up the textbook. I'll walk you to where it's going. And then you'll see it, the physical structures inside the brain. You know, why does Mother Nature give you this thing that's called the corpora quadrigemina, right? It's because you have two ears that need to coordinate with two eyes so that when your reflexes are working together, you know, you actually can see and hear whatever it is that's stimulating that actual, hopefully, response. Okay. Do you have any other questions before I sign off for the evening?
I don't think so. That puts us right through to chapter 14, right?
That actually gets us into chapter 17 there, believe it or not. Okay, so point being is that tomorrow's lab, I'm going to work primarily on 12, 13, and 14. And at the end, I'll introduce 17 so people can do well on the practical.
Okay.
Okay. So you have yourself a great night, and I will see you tomorrow evening. You found the recording stuff, right?
Yes, I did. I've been watching it.
Fantastic. Okay. Well, you have a great night, and I will see you tomorrow. Okay.
Thank you.
Of course. Bye-bye now.